Dr. Becher, who was recruited to Mount Sinai last year as a professor of pediatrics and oncological sciences, a pediatric neuro-oncologist, as the chief of the division of pediatric hematology-oncology. He completed his BA at University of Pennsylvania, followed by MD at Johns Hopkins, and then a pediatrics residency at Children's National in Washington, D.C., before moving to New York and completing a Hem-Onc fellowship and Neuro-Onc fellowship at Memorial Sloan Kettering Cancer Center. Dr. Becher has been a researcher now for a number of years and has been focusing his energy on developing mouse models for DIPG. And he's really been a leader in that field and has made some of the more important discoveries that we've seen in the last decade or so in evaluating the driving mutations underlying certain DIPGs. I had the pleasure of collaborating with him a little bit when I was a resident and doing some DIPG research. He's a great collaborator. He's a good friend and colleague. And we're looking forward to hearing his talk about the importance of histones in midline glioma today.
Thanks for the kind words, Peter. Great to be here. I've been here now 6 months, slowly getting settled. My mice finally arrived at Sinai after a few months in Boston at quarantine. So we're eager to get going here and move the science forward. So let me just share my slides if I can find. Second. Hold on 1 second, still having trying to find my presentation. Yeah. Hold on. Oh, it's under my PowerPoint folder. I see. Let me move that over. Try again. Okay, here we go. Are you able to see it?
Yep, there we go. You can see it.
Okay, great. Actually, it's a funny format that I'm not used to, but hopefully it will go smoothly. Okay, great. Well, again, thanks again for having me here. So I've been as a fellow at MSK, I was taking care of some children with DIPG, and this is around 2006, 2007. And I realized very quickly that at the time, all the clinical trials at MSK for DIPG were like farnesyl transferase inhibitors. And at the time, it became very clear that basically everyone was just, there were no models for DIPG way back then. And all the clinical trials were just based on adult glioma models. And I thought that was odd. And so I, basically serendipitously decided to focus on this particular tumor. At a time, again, 2007, 2008, I may have been like one of the first DIPG researchers. Little did I know that like now it would be like a very crowded field. But back then, almost no one was really working on it. And I think, and you'll see that the histone mutations that were discovered a decade ago, exactly in 2012, are one of the reasons why the field is actually extremely crowded now. It just brought in like all these basic scientists, chromatin gurus, trying to see why, how do we crack this puzzling, challenging tumor. And so, again, I think the kind of like the big picture aspect of this is that the histone mutations confer a really poor prognosis. In fact, at least for DIPG, any child that I've taken care of over the years with a K27M histone mutation is no longer alive. So I think, basically like it's a challenging tumor. We don't know, as you'll see from my data, again, this is about a decade worth of work or even longer. You'll see that we still don't know how to target it, and we're just getting a glimpse of how complex it is. And so I'll show you some of that data. So I guess I'll start with just sort of like a kind of make sure that everyone's on the same page. I actually I've been here 6 months and I haven't seen a patient with DIPG here at Sinai yet, but my goal with Peter and Sadi to change that over time and eventually have patients with DIPG come to Sinai as a destination. So that's my long-term goal for the program. So again, these tumors are, they've sort of went also a change in name. So historically we called them all DIPG or diffuse intrinsic pontine glioma. But now when I submit papers, the reviewers always ask us to change it to diffuse midline gliomas. And this just highlights how much the field has grown, is because the WHO has included diffuse midline gliomas with the K27M mutation as a distinct category. And so I think basically every pathology program across the country or the world has to test for the K27M mutation almost in my opinion, it should be done in all patients, but I think at least in midline gliomas. So again, usually this presents in first graders. You can see here with the sagittal image that these tumors mainly don't enhance with gadolinium. This is a T1 with contrast. And again, the survival is still less than 1 year from diagnosis in 2022. In the United States, there's about 250 kids per year that get it. The classic triad of presentation is cerebellar sign, long trach signs, and cranial nerve palsies. Usually a 6th nerve palsy is the most common cranial nerve abnormality. And so why is this tumor so challenging? Why has there been no progress? I think, one of the challenging aspects about it is that it's not resectable. So I think, again, when I started working on this 15 years ago, even biopsies were considered risky, but through the work of some pioneering neurosurgeons like Stephanie Puget in France, it was shown very clearly that a biopsy of the pons can be done safely. And I think maybe the complication rate is extremely rare, maybe like 1%. So again, even I still get emails from families, newly diagnosed families with DIPG, and ultimately the first question they ask is whether the biopsy should be done. And I still recommend it with caution, but I still think it's like the way to move forward. But just because, like, as someone who's been in the field, I've seen occasionally, again, it's 1 out of 100. I've seen complications from a biopsy where you get like a hemorrhage, and that could be sometimes catastrophic. So, but again, it's really rare. So I still advocate for a biopsy, because if we don't know exactly the genetics of it, then I think we will never get to a cure. Again, I think the other challenging thing, which you saw in the previous slide with the MRI, is that the blood-brain barrier is intact. And so this is why we have a tumor here in 2022, where the standard of care is focal radiation, and that hasn't changed. And these tumors are highly infiltrative. So I think by the time they present, they're already, the tumor cells are already outside the ponds. And so where are we today? And this is sort of like my big view picture of where the field is. So again, the standard of care, focal radiation, has not changed since the 1960s. We've done clinical trial one after another, and they've all failed with various agents, molecular targeted agents, chemotherapy. I think about a decade ago, I could get away with just saying, well, because we were just using the wrong models. Most of the preclinical data way back like a decade ago was using U87 cell lines. And so that's why we didn't get anywhere. But now we're actually using DAPG cell lines. And I look at the clinical trials that are available, and I still am not confident that any of them are actually going to make like a dramatic difference. And so I think, these tumors are just really challenging. And I think we need like some more basic science and some creativity to try to move the field forward. I think a lot of the families that have been affected by this tumor have been really great advocates. They start their own foundations. They've been demanding biopsies. They've been doing autopsies, asking for autopsies on their children. And I think that all that has really led us to have a greater understanding of the biology of this tumor. And so I am cautiously optimistic that in the next decade or so, there will be some treatments that will get approved and will move the field forward. I think some of the challenges in the field is there's a lot of desperation on the side of the families to get clinical trials going. And so I think sometimes the bar for moving something into a clinical trial is really low. As an example, I think CAR T cells, I think are really a hot area in DAPG. Some of you may have seen like a recent Nature paper with targeting GD2 as a CAR T cell. But again, I think it's hard for me to interpret that paper to say that it worked. I think it seemed very intense. And I think we didn't necessarily know why it didn't work as better than it did. And I think part of the challenge is that some of the preclinical data for that particular testing was done in an immunodeficient models. And I think what we're facing with is like, which are the best models? Immunodeficient models are nice because you can use the actual human cells from the patient. But if you're testing immunotherapy and the mouse has like no immune system, I think it's hard to really know what's, if that's like valid results. And so, again, when I started working on this tumor in 2009, we almost knew nothing about the genetic drivers. I've been fortunate to be part of some of these discoveries of these genetic drivers. Again, the histone mutation is sort of like the big elephant in the room. This is a true story, but actually when I was at MSK and I collected 12 of these samples before the histone mutations were described by Nada Jabato and Susie Baker. And I actually had this 12 specimens, and 11 of them had the histone mutation, and I gave them to the MSK core facility to sequence, and they missed the histone mutation because of histone mutations, and I'll talk about it in a couple of slides. There's so many histone genes that are so similar. Sometimes the sequencing machines think of them as like dubious, like there's too many genes that are histone genes, and so the algorithm at MSK for the sequencing machine just like tossed those sequences out. So actually, like we had the data that the histone mutations were important drivers, like 11 out of 12 had, but we didn't know that we had this mutation because the bioinformatics wasn't up to speed yet. Also in 2009, there were no models. Today we have like hundreds of models, both genetic models and xenograft models. So I think, there's a lot of excitement and potential in the coming decade. Again, just a little bit of background. I think one remarkable thing about the histone mutation is that they're region specific. And so the K27M is primarily in the midline, although you'll read case report that the K27M mutation can be seen in rare tumors in the cerebral cortex. So I personally advocate that every patient with a tumor in the brain should get a K27M histone mutation as part of the workup. Just because sometimes you get surprised and, the tumor, even in the cerebral cortex, can have a K27M mutation. The G34RV mutations, they're more rare. They also have a poor prognosis. And they've been less studied. And so I'm actually not going to show you any data today, but we have recently made a model for this tumor as well. And so let me just go a little bit on background about the histone. So again, we have 30 copies of histone 3 in each cell in our body. And only 1 out of 30 copies is mutant. So it's a heterozygous mutation in one histone gene, most commonly the H3F3A. And so I think the remarkable thing is that 1 out of 30 is enough to cause a phenotype. And again, when we've done mass spec, when we collaborated with David Alice, about 5% on average, but there's a range, 3 to 10% maybe, of the total histone 3 pool by protein analysis, mass spec, is mutant. So it's a small percentage, but it's enough to cause a phenotype. And just as a reminder, so histone 3 is part of the nucleosome, right? And the nucleosome is comprised of 2 H2As, 2 H2Bs, 2 H3s, and 2 H4s. It's the nucleosome, it's like helps regulate, it helps package the genetic code inside the cell. And you can see here that the mutations are in an end tail of H3. And this is like a really important region that regulates gene transcription. And so, I think that an important advance in the field was when the Stefan Pfister group in Germany, they did unsupervised analysis of about 200 glioblastomas. And this is unsupervised analysis. So, they use both pediatrics and adults. And lo and behold, if you cluster these in an unsupervised way, you get 6 groups. And the histone tumors cluster separately from each other. So you have the K27M group that has a unique pattern. You have the G34R group that has a unique pattern. The IDH tumors have a unique pattern. And so again, this is this paper was like maybe like a decade ago, but the group in Germany, they have really pushed this as a diagnostic pattern. And so now in select sites across the US and the world, if you have a tumor in a child that you're not sure what it is, you can send the sample to get methylation analysis. We don't have one at Sinai yet. I was told by the chair of pathology that this is something that we're hoping to purchase. Now, if we have a tumor that we're not sure what it is, we would send it to NYU because they have a machine. But I think this is like a great diagnostic tool because, it seems like the DNA methylation pattern nicely corresponds with some of these genetic groups. And so again, so how does the histone mutation cause tumors? I think that's sort of like a question that was actually figured out really quickly. So what happens is that the histone mutation interferes with the function of this important complex called PRC2, which stands for the polycomb repressive complex 2. The active enzyme of this complex is called EZH2, enhancer of zest homolog 2. This is important because actually EZH2 inhibitors are actually FDA approved to treat some cancers like lymphoma. And it's basically works best in tumors that have activating mutations in EZH2. But there's actually some literature in DIPG that says that EZH2 inhibitors may work for DIPG. I think it's controversial. We didn't have great success in our models so far with EZH2 inhibitors, but we've seen that it works in some of the human models. I don't know why we get different results with different models. I think that's one of the challenges in the field in general. But the K27M interferes with this enzymatic activity, which this complex adds methyl groups to H3K27, and H3K27 trimethylation, when it's next to a nearby gene, it correlates with gene repression. So it's thought to play a role in turning genes off during development. So then every cell is expressing the correct genes for it to become like a neuron or an oligodendrocyte or an astrocyte. And so actually when Dave Alice and Peter Lewis started working on this, and I was at Duke, they emailed me to collaborate. And again, they were part of this seminal paper in Science in 2013, where basically, if you can look at this Western blot, I think it tells the whole story nicely. If you take HEC293 cells, and then you add these different histone constructs, if you add the K27M, you get a drop in trimethylation of H3K27 trimethylation, dimethylation. There's a little bit of an effect on the monomethylation. So this mutation, when it's expressed exogenously, can reduce global levels of H3K27 trimethylation. And so this is sort of like the hallmark of the mutation. I don't have time to go into the biochemistry, but the group from David Alice's lab showed nicely that the K27M inhibits this complex in these nucleosomes, and this has a profound effect on the cell. And so it doesn't work. The mutation, even if it's in this nucleosome, it not only inhibits trimethylation of H3K27 in the nearby histone 3, but it also affects nearby nucleosomes. So there's like this global effect. This is like just a cartoon to kind of summarize what's going on with this K27M mutation. Again, it's a subject of an intense study. We still don't know how to target it. It's found in like 90% of the APGs. It causes a global reduction in H3K27 trimethylation, but also focal gains in H3K27 trimethylation. And then it also causes DNA hypomethylation. I'm going to show you some of our work showing how it causes focal gain in H3K27 trimethylation and why that's important. Sort of like the way I think about cancer has to do with like an important mentor when I was at Hopkins as a medical student, Bert Vogelstein. Some of you may know him. He sort of spent like years studying colorectal cancer, and he sort of teased out in a stepwise fashion how genetic alterations are acquired over time and eventually go from like, just like a benign small adenoma to a carcinoma. And so the way I think about DIPG is sort of similar. The histone mutation, by far, most of the data suggests this is an initiating event. It's the first mutation. Now, sometimes K27M tumors can also be found in low-grade tumors. And I think in those situations, the subsequent mutation is like a low-grade genetic event, like a BRAF V600E. So if you start with a K27M and then you get a BRAF V600E, those tumors tend to be more indolent and they're less aggressive. They're still probably more aggressive if the histone mutation wasn't there, if it was just a BRAF V600E, but it's not as bad as if we go the other way where you acquire either a TP53 mutation or a PPM1D mutation. I didn't get a chance to go over this, but the PPM1D is a phosphatase gene, and there is truncating mutations that results in overexpression of PPM1D, and PPM1D opposes TP53. So when there's too much phosphatase around, it inactivates P53. After you get these TP53 and PPM1D mutations, then you get RTK alterations, and depending on which one, it affects how aggressive the tumor is. So again, I think it's also a stepwise process, and I think if you go in this way, we're not smart enough yet to like get around that. And so what kind of questions can you answer with a genetic model? I mean, we sort of spend a lot of time trying to combine different genetic events to make a DIPG. I'll show you some new data that has not been published yet, that is hopefully will be published maybe next month, about how the cell of origin actually has a huge effect on whether how the K27M is oncogenic. Like it has a different mechanism depending on the cell of origin. We're really excited about this data. I think it shows how complex this mutation is. I mean, functional genomics it just means that you can we can express whatever gene we want in a cell in the brain and see what it does. So I think it's important to study things in vivo. So that's sort of like the focus of what my lab has been doing. And then we also evaluate novel therapies. Again, in general, we found it almost impossible to cure our mouse model. Even radiation has a pretty weak effect. So I truly believe, although maybe I'm biased because of, because I work on this model, I truly believe that if we can actually cure the mouse model, we can cure the patients. But at this point, we're not able to cure the mouse model at all. Nothing really cures it. So the system that I use is called the RCAS-TVA system. I think Dolores is here as well. So you probably have heard some of this work or similar work from Dolores, who's studying the myeloid cells. And so I think, now we have really a lot of RCAS-TVA expertise. again, I think it's just like a clever way to model cancer in the brain that Eric Holland pioneered. It allows us to deliver whatever gene we want with some limitations on the size of the gene into specific cell populations. And my lab has been using mainly Nestin, but I'm also going to show you some data with ALEC2 and GFAP as cells of origin. We also use PAX3, but I'm not showing you that data today. So here's a schematic. So basically, we make the ARCAS vector that we want to study. We put whatever gene we want. And then we use virus-producing cells. And then we inject them into the pups when they're P3 to P5. And we use a Hamilton syringe. And then we wait about 6 weeks, and then we get a tumor. And so before the histone mutations was discovered, we are mainly making gliomas in the mouse by just turning on PDGF signaling. So if you just turn on PDGF signaling by itself with either the PDGF-B ligand or PDGF-A ligand, you get gliomas. And they're mainly low grade if you're doing it in the brainstem. If you want to get higher grade tumors, you have to add a tumor suppressor like deletion of P53 or deletion of INC4A-ARF. And then you can get more aggressive tumors more quickly. As you can see here from survival curves, that we can get a pretty aggressive tumor in a brainstem driven by just two things, PDGF-B and P53 loss in 40 days. So I think it's a very aggressive model with like pseudopalisade necrosis, microvascular proliferation that is basically, I think at this point, incurable with today's tools. And so when we first made the model, again, this was work that I was doing before we knew about the histone mutation. But if you look at it, the model that we made even back then in 2010, which was just PDGF-B and INC4A-ARF loss, looks remarkably similar to the human tumors that have the histone mutation. So again, I think it highlights that, and I think this is serendipitous that we sort of focus on INC4A-ARF loss because I'll show you that the K27M actually inhibits INC4A. And so I think maybe that's why the initial model wasn't so bad. Again, there's a lot of complexity to DAPG. There is two flavors of the K27M mutation. There's a 3.3 and a 3.1. I didn't have time to go into this, but just in brief, the 3.3 is a non-canonical histone and a 3.1 is a canonical histone. The only difference between those two genes, it's about 5 amino acids, is that the 3.1 gets incorporated into the chromatin only during S phase, and the 3.3 can be incorporated into the chromatin independent of S phase. There's this remarkable thing that we don't fully understand, why the 3.1 K27M partners up with ACVR1 and the 3.3 K27M partners up with PDGFRA. I think it's like an interesting puzzle. So you can get different flavors. The red colored genetic drivers are more aggressive than the orange colored genetic drivers. And so when the histone mutation was first described, a decade ago, we did the simple experiment of just overexpressing it in the neonatal mouse to see what happens. And basically, we noticed that you don't get any tumors, even with 53 loss, and you get these preneoplastic clusters that express K27M, but they're non-proliferating. So if you look here with the double immunofluorescence, the green cells are the histone mutant, and the red cells are PCNA positive. And you can see that these cell populations are non-overlapping, meaning that the K27M cells are not proliferating. And I think this touches into our theory of what's going on, is that the K27M works best if the cells are proliferating. But if the cells are not proliferating, then the K27M is not that helpful. And so to make the model with the K27M, we went back to using the PDGF system because we knew that that's enough to make a glioma on its own. And so how do we study the histone mutation in the context of PDGF signaling? We can study it with P53 wild-type tumors, which are more indolent, or we can study them with P53 deletion, which is a more aggressive model. And so we did a simple experiment. You just turn on PDGF signaling in the mouse brainstem, in the nestin cells, and then you can either add empty vector, the wild type histone, or a mutant histone. And as you can see with this cartoon over here, if you add the histone mutation, now half of the tumors become high grade. And if you do EDU labeling, you get an increase in proliferation. So this shows that the K27M cooperates with PDGF signaling to increase tumor grade and proliferation in vivo. Notice that in this P53 wild type model, the survival curve is relatively flat, meaning that most of the mice survive just fine, even if they have a low grade tumor in the brainstem. These tumors don't progress that much. So again, this is the low grade model that we use, which is P53 wild type. If you want to get an aggressive model, then we have to use P53 deletion. And then in that situation, you see here that the red line, which is with the K27M, is a little bit more steep than the black line. And this is significant, but it required a lot of mice. So it tells you that the effect is pretty modest. So the K27M, when you use PDGFB and P53 loss, you get an acceleration of tumorigenesis, but it's pretty modest. You do get the drop in methylation, which is sort of like as a positive control that you actually did what you think you did. Here's some pictures comparing the mouse data to the human data. The human data, you can see here on the left panel, shows, again, the wild type DAPG has a lot of H3 K27 trimethylation, and the K27M mutant has very little H3 K27 trimethylation. We quantify that over here. In a mouse model, there's not as much H3 K27 trimethylation. So it's a little bit different, and I don't know that we fully understand why that is. And then if you add the K27M, you still get a more further drop in trimethylation, but you started with lower amounts. So I think it could be like a species differences between mouse and human. So then we wanted to kind of study this in vitro. So you can see here that if you take cell lines from either an H3 wild type tumor or a K27M tumor, the K27M tumors proliferate faster. So, you can see there's a higher, this is a BRDU labeling. We sorted for GFP, so you're only looking at tumor cells. If you do a cell cycle analysis, you can see that the K27M cells are proliferating faster. Next thing we did was we did RNA-Seq. We wanted to identify, so what are the target genes of the K27M? Like, how is it causing tumor formation? And I think, as you would expect from what I said earlier, that if you have the K27M, you get a drop in H3 K27 trimethylation. And so you upregulate a lot of genes. But we actually were interested about these genes that are repressed by the K27M mutation. And so we studied this in further detail. And so you can see here that the K27M mutation, one of the locus that showed up on the RNA-Seq was a CDKN2A locus. So you can see here that whether you take tumor neurospheres or ex vivo progenitors, the K27M mutation reduces P16, which is INK4A. It's a tumor suppressor that binds CDK6. So it's an endogenous inhibitor of CDK4/6. And if you have reduced levels of it, then the cells can cycle faster. We wanted to understand the mechanism of this. How is this happening? And so we noticed that there is extra H3 K27 trimethylation at the P16 promoter. And so this is interesting because I told you before that the histone mutation mainly reduces H3 K27 trimethylation. And we don't understand how it's happening that there is extra H3 K27 trimethylation. I think this is a conundrum for the field that hopefully will be sorted out by the basic scientists. But basically, if you look at the P16 promoter, there's extra H3 K27 trimethylation in the K27M cells. And if you treat with an EZH2 inhibitor, you do get the levels to go down. So we were like excited, okay, let's treat with an EZH2 inhibitor and see if that helps as a therapy. Unfortunately, there is no effect. So the EZH2 inhibitor doesn't do anything whether you treat with the H3 wild type cells or H3.3 K27M cells. Here you can see here that there's no upregulation of P16 transcript. So again, this shows you that these tumors are really complicated, and even though we think that the P16 is repressed by the K27M, treating with an EZH2 inhibitor doesn't really solve the problem. So we ended up looking at DNA methylation, and it's a long story short, but there is extra DNA methylation at the P16 promoter. And if you treat with a decitabine, a DNA methyltransferase inhibitor, you can reduce the DNA methylation, you can rescue the P16 transcript. The problem with DNA methyltransferase inhibitors is that they affect DNA methylation, not just at the P16 promoter where we want them to work, but throughout the whole genome. And so, this is going to be probably like a toxic therapy, but I think, it did seem to restore P16 levels, and it does have anti-tumor effects in our model. So now let's going back to the cell of origin, because we got some really interesting results that will be published shortly, hopefully. So we noticed that if you analyze the human tumors, you see that most of the tumor cells are OPC-like. This was a paper in Science by the Boston group, showing that if you look at the IPG and you do single cell analysis, all the cells have an OPC-like signature. So then we were like, well, maybe we used the wrong model. Maybe we used the wrong cell of origin to model the IPG. What if we looked at ALK2 as a cell of origin instead of nestin? And so we repeated all the experiments with ALK2 as a cell of origin. We also looked at them in the neonatal brainstem with collaboration with Dolores's lab, and found out that ALK2 cells co-label nicely with SOX2. This was nice triple immunofluorescence done by a postdoc in Dolores's lab, Gonzalo. And we see is that there's a lot of overlap between ALK2 and SOX2. So in the neonatal brainstem, a lot of the ALK2 cells are OPCs, or oligodendrocytes progenitor cells. So then we said, okay, so let's do the same experiments that I showed you before with the nestin model. And let's see if the K27M will make a tumor if we initiate tumorigenesis in ALK2 progenitors. And to our surprise, the K27M doesn't do anything if you overexpress it with P53 loss in ALK2 progenitors. So you get no tumors. And then if you look at P53 wild type mice with PDGFB, the K27M doesn't have a significant effect on survival. But then if you look at tumor grade, and this was reviewed by a neuropathologist, Dan Bratt at Northwestern, you can see that the K27M does make half of the tumors go to higher grade. This is quantified here. So again, the K27M does have an effect in this model, and but it's sort of similar to what we saw with the nested model. So then we looked at P53 heterozygote mice. We wanted to, again, we're trying really hard to find, to study this K27M mutation in a different cell of origin. And in P53 heterozygous mice, we don't see an effect of the K27M mutation. This is sort of like a summary for all these experiments. You can see here, the red line is a P53 heterozygote model. And the black and blue line are the P53 null model. So you can see that the P53 is doing a whole lot more than the histone mutation. And so, in our model, I don't know if it's a mouse thing or it's also true in the human disease, but in the mouse modeling, P53 has much more of a dramatic effect on tumor latency than the histone mutation. So then we did RNA-Seq. And so if you look at the PDGFB model, what we found out was that you get very few genes that are significantly differentially expressed with the K27M. However, there is enrichment for oligodendroglial differentiation with the K27M. So this data suggests that the K27M mutation pushes cells towards oligodendroglial differentiation. Another thing that we did was also compare PDGF-A to PDGF-B. If you look in this graph on the right, the blue line is PDGF-A and the black line is PDGF-B. So again, we are seeing that the PDGF-A versus B ligand is much more important than the histone mutation. On the left-hand side, this is the effect of the histone mutation in the PDGFA model. Here's the effect of the histone mutation in the PDGFB model, basically like very subtle effects. But the PDGFB versus A has a much more dramatic effect. One thing that was really remarkable that I'm really excited about is if you stain for K27M using an antibody, there's different levels of K27M depending on the model that you use. The nested model has a lot more K27M cells. This is quantified here. GFP is because we also have a GFP tagged histone. So the GFP results is a confirmation of the K27M IHC. But basically, you can see here that in the PDGFA model, in nestin as a cell of origin, all of the cells are K27M positive. On the other hand, the ALK2 model, if you use PDGFB and ALK2, only like 30 to 40% of the cells are K27M positive. This tells us that if nestin is a cell of origin and we use PDGFA as a ligand, K27M is very important. In general, the more important a gene is in tumor genesis, the more highly expressed it is in the model, because the model is sort of like a genetic experiment. If a gene is really important, it gets incorporated and it's expressed in high levels. If it doesn't have any selection force for it, then it gets expressed in low levels. So this was, again, this kind of validates some of the results that I'm going to show you in a second. The person that did this work is Yusuke Tomita. He's an MD PhD neurosurgeon from Japan that spent 2 years in my lab. He was basically like, he worked really hard. He was a machine. Here are all the tumors he made in the mice. So again, kind of like to recap this part, because I think it's really important. And again, this is all unpublished, but I think it hopefully will be published next month. The K27M has very minor phenotypic effects when ALK2 is a cell of origin, even though you get a drop in H3K27 methylation. When nestin is a cell of origin, the K27M has dramatic effect. There is also a corresponding effect on proliferation. And so you've seen like two different cells of origin and very dramatic differences in the K27M. One thing that we did was we did Rna-Seq and then we did GSEA analysis. And it's interesting, even though the K27M doesn't do a whole lot on survival in the ALK2 model, it does prolong EMT and angiogenesis it promotes EMT and angiogenesis in a robust way. And so I think the K27M is doing stuff in different cells, but the mechanism is different depending on the cell because the chromatin is different. Here, this is going back to the nestin model. Again, we're seeing that the K27M promotes myelination and actually inhibits EMT and angiogenesis. So what we're seeing is, and this is highlighted on this graph right here, this is GSE analysis, is that the K27M can have opposite effects depending on the cell of origin. And I think it just really tells you that, this kind of mutations that affect epigenetics like H3K27 methylation or all these histone marks, depending on the cell that the K27M infects or is expressed in, it's going to have completely different effects. It all depends on the chromatin. So in conclusion for this part, the K27M mutation accelerates gliomagenesis both in P53 wild type and P53 null settings when nestin progenitors are the cell of origin. The K27M does not accelerate gliomagenesis if PAX3 is a cell of origin. I didn't have time to show you this data, but it's published. The K27M does not accelerate PDGFA-driven gliomagenesis in P53 loss setting when ALK2 is a cell of origin. And the effect with PDGFB was very small. ALK2 and nestin don't overlap much in the neonatal ponds, and most ALK2 cells express SOX2, consistent with ALK2 cells being OPCs in development in this kind of postnatal time point of P3 to P5. Our results show that whether you use PDGFB or PDGFA in the model has a much more dramatic effect, and P53 status has much more dramatic effect than the histone mutation. I also showed you that the K27M can affect lineage and it promotes an oligodendroglial cell fate in certain contexts. And its effect on angiogenesis and even EMT are cell of origin dependent. And the expression of K27M varies by the cell of origin and by the choice of ligand you use. So again, just kind of summarize here, basically we analyze human tumors, whatever the genetic drivers are, we kind of put them in the mice and make models and then test therapies. In the last few minutes, I want to show you some work that we've recently submitted for publication that I think is pretty interesting. I've always been intrigued by PFA ependymoma. And it's a nice comparison to DIPG because PFA ependymoma, the reason that I'll be very interesting in a second, the reason is because PFA ependymoma doesn't do so bad. The 10-year survival is 50%, and because you can resect most of it. So neurosurgeons are critical in PFA ependymoma management. You know, if you can resect almost all of it, that gives the patient a much better chance of long-term survival. The reason that I think it's important to compare the two is because in the recent years, the DIPG is characterized by the K27M histone mutations. And in the last few years, it turns out that PFA ependymoma have a gene called EZHIP or catacomb or CXORF67, which is just like a K27M. So here we have two different tumors in different locations, and both of them have the same kind of K27M mechanism, but one has a 10-year survival of 50%, and one has a 10-year survival of like 0 and a 1% survival at 5 years. So very different outcome, even though the biology seems to be similar. And I think it really highlights that the reason they were doing poorly in DIPG, in my opinion, is the fact that these tumors are infiltrating in the pond, so we can't take them out. And also some of the other associated genetic alterations are different. So P53 mutations are not commonly seen in PFA ependymoma, and they're commonly seen in DIPG. And I think that plays a role in why DIPG is practically a death sentence. With PFA ependymoma, there's a very good chance that you can have, you can potentially beat it. Here's some data showing that PFA ependymomas also have reduced H3K27 trimethylation. You can see that here. PFB ependymoma, on the other hand, has a lot of H3K27 trimethylation. So you can see the contrast. Both of these are in the posterior fossa, but there's a dramatic effect on H3K27 trimethylation. Again, this is the work of other people, but my lab has subsequently made a PFA ependymoma model that I'm going to show you in a couple of slides. But again, the reason we figured this out is, us as the brain tumor community, is people realize that CXORF67 and H3K27M have mutually exclusive mutations in PFA ependymoma. And so nobody knew what CXORF67 does, but people expressed it, and then they noted that it reduces H3K27 trimethylation. So this slide is just to show you that even ependymoma needs to be molecularly characterized. I'm gonna, so basically this is a nice schematic. So this new gene, catacomb, it's not expressed in the normal human brain. It's only expressed in germ cells. So like in the testes, it's really important, but it's not expressed, and it's aberrantly expressed in PFA ependymoma. And it's really cool that it has a K27M sequence that inhibits PRC2. And so my lab, when we saw this data, we're like, well, let's try to make a genetic model for PFA ependymoma because nobody has made one yet. And we, to make our study rigorous, we made I'm going to call this gene catacomb, but it also is called EZHIP or CXORF67, but catacomb is one of the names it has. So catacomb wild type is the same as the K27M. And we also made an M406K mutant. So this is a loss of function mutant where you lose the K27M activity as a control, just to show you that this histone mutation is really important, or this catacomb gene. So if you look at this data, look at the survival curve on the top left. The blue line is catacomb wild type with P53 loss and PDGFA. The red line is that loss of function mutant. So you can see that there's a dramatic effect on survival just by this having a wild type catacomb versus a mutant catacomb. And you can see here that if you look at the histology, we do get ependymoma histology. One thing that's really cool is if you look, we had Dan Bratt at Northwestern grade these tumors, and the wild type tumors have significantly reduced ependymal differentiation than the loss of function mutant. So what I'm saying is that the catacomb gene, it's accelerating tumor genesis. It's also inhibiting ependymal differentiation, and it's reducing H3K27 trimethylation. So I think it's, I think this is pretty cool results that we think it really shows that these epigenetic genes, they mainly inhibit differentiation. And PFA ependymoma cells, not all the tumor cells in a PFA ependymoma are ependymal. There's a lot of stem-like cells. And I think as the tumors come back, this was shown by Mariella Philbin, when these PFA ependymomas come back, they become more stem-like. They're more undifferentiated and they're harder to treat. We also did the same experiments with GFAP as a cell of origin, and we got similar results. So, the only difference was that when you make GFAP as a cell of origin, now the catacomb doesn't have any effect on ependymal differentiation. That was the only difference. So we think the Nestin is probably the better cell of origin for PFA ependymoma. But, we still get some tumors that look like PFA ependymoma. And I think most people think that GFAP is, it's like a radial glial marker. So people think that may be like a better cell of origin. So I think the jury is still out. What's the best cell of origin for PFA ependymoma? That was just our initial efforts to make the model. Also, we saw that some of these tumors have ependymal lining. And the staining here is for catacomb. It's very sporadic. It's not in every cell. We did some RNA-Seq and basically confirmed that the catacomb wild type affects H3K27 trimethylation and the signature associated with that. We also saw something that's really cool is that there's a lot of ependymal genes that are upregulated by catacomb wild type. So it seems like this gene, catacomb wild type, can turn on ependymal differentiation, and it's more pronounced when Nestin is the cell of origin. In fact, if you compare Nestin as a cell of origin with the loss of function mutant between the tumors that we got with the Nestin as a cell of origin versus GFAP as a cell of origin, the tumors that were initiated in Nestin cell of origin are more ependymal. They express higher levels of ependymal genes like aquaporin. Here's a picture of one of these tumors. You can see that the brown staining is this catacomb gene, and you can see that it's expressed not in every cell, and maybe more in these more high-grade areas. So in conclusion for this part, basically the cell of origin impacts the transcriptome more than the catacomb activity. With the RNA-Seq, we showed you that the K27M or the catacomb wild type significantly accelerate tumor genesis in Nestin progenitors relative to the loss of function mutant. The catacomb mutant tumors have significantly increased ependymal differentiation. We showed that you do get inhibition of PRCA2 activity by GSEA analysis. And we think the Nestin is probably a more accurate cell of origin just because even the catacomb mutant tumors, they just had more ependymal genes. And I showed you that the catacomb wild type versus mutant, there's reduced levels of ependymal differentiation. I only have 4 minutes left, so I'm going to just zoom through this last part. The last thing I wanted to show you is that if you make genetically identical tumors and then you do dynamic contrast MRI, the tumor location, so whether a tumor is made in the cortex or in the brainstem, affects the blood-brain barrier. So you can see here, this is something that we published a few years back, but it really highlights that the blood-brain barrier for a tumor is completely affected by tumor location, and that's why brainstem tumors have a more closed blood-brain barrier and they're harder to treat. I mean, I've had a few patients over the years that have a GBM, but it's in the cerebral cortex. And if it's a young child and you do a gross total resection, that child with the GBM can have a long-term survival and be cured. So I think the tumor location is really important. Lastly, I just wanted to put a plug in about hopefully we can get a methylation machine. This is like the methylation results, DNA methylation that you can use to categorize tumors. So we're moving away from the optical era to the molecular era. This was like a recent paper by Stefan Pfister in Cancer Discovery. And I think it's really important to do a molecular analysis of the tumors, especially, I think in general, but particularly in pediatric brain tumors. So with the last couple minutes, I have my new lab here at Sinai. Yi and Millie are my first two lab members. I'm grateful for them. We're just getting the lab set up. These are all my former lab members, my collaborators, my funding sources. particular, Praveen and Dolores have been close collaborators, and I'm excited to be at Sinai, and hopefully maybe I will be able to collaborate with some of you. Again, thanks for your attention. Happy to take any questions.
Oren, great talk. Kostas here.
Thanks, Kostas.
You gave your presentation well while drinking your Starbucks coffee, so good job. Anyhow, so when we go back to the glioma stem cell story, it's one that really got a lot of attention early, and I think your data, your work has shown kind of the nestin cell of origin. All roads lead to the nestin cell of origin, it seems like, right now for DIPG. I mean, is that a accurate assessment, or is that?
Yeah, no, I think so. I think these tumors arise from nestin cells, but they do become OPCs once they get the K27M mutation, and then all these other like PDGF signaling. Then they become OPCs, but they started out as stem cells.
Right. So that's the point. So is this, are we now supporting the stem cell hypothesis? Is this the nestin stem cell origin of DIPG tumors that kind of lead to the tumor genesis and just therapy resistance of these tumors? I mean, I know the blood-brain barrier in these tumors in particular is especially difficult because of not much enhancement and just pretty much an intact blood-brain barrier. But is that what you're thinking in terms of how the DIPG stories unfolding?
I mean, I think that the plasticity of the cells, the tumor cells, they're so plastic that they're hard to cure for that reason. That's why they're radiation resistant. So I think, yes, the stem cell, the plasticity of the cells, that they can go into different lineages, I think that's what makes them really hard to treat. And that's why we're like, even in the adult GBM world, we've sort of failed these patients. I mean, it's very hard to cure them. So I think, that is a challenge that we need to overcome. These cells are really tough to get rid of.
Nice job.
Thank you.
Oren, I had a quick question for you. Great talk, and thank you so much for bringing your expertise to Mount Sinai, and we look forward to collaborating with you. Comment about your biopsy point in the DIPGs. I think it's critical, and I do think it's safe. We've done the number here prior to you getting here, and with robotics, it's a safe trajectory coming from above is the way that I like to do it, and I think it's absolutely fine and critical. I had a patient in the era before you got here that had a midbrain tectal glioma that was not indolent, and I did a biopsy of this, and it had K27M positivity. She's gone on, and she illustrates that point that you made about how some of these are low grade, and she's, I think, 7 years out now and doing absolutely fine. So what is it about this midbrain that is 3 millimeters away from the pons that makes it so different? I just don't, and Fred Epstein in the 90s talked about this, how the midbrain was benign, the pons was malignant, and the medulla was somewhere in between.
Yeah, no, that's a great question. I think we don't know the answer to that. I think those are good points. Thanks for mentioning that you've biopsied the APG. From now on, if I get a new DIPG family asking for my opinion, I'm going to refer to you or Peter.
Yeah,
I mean, we can get them here.
With Rosa, it's absolutely no problem. I've done these on a number of occasions, both here and at West, and it's very smooth. And when you come from above and you're following the tracks going rostrocaudally, it seems to be quite safe.
Okay, well, good to hear. But yeah, I mean, I think you bring up an excellent point. Like, we don't know why the pons is so more challenging than the midbrain or medulla. I think it's a great question that we can keep thinking about.
Thank you so much for the talk. That was great. I just had a quick question. In the nestin models, did you also see, I may have missed it, did you also see that impact when you had the P53 heterozygote that you saw in your other cell line in terms of the importance of the P53?
No, I think the P53 heterozygote data was like a bust. It didn't, we didn't get a significant effect with the P53 heterozygote model.
But you use that to confirm, or to demonstrate the importance of P53, or showing that was having a significant impact versus the methylation status. Is that right?
Yeah. So our data, and again, I think, everyone, there's been a lot of bias that the histone mutation is like the most important target for DIPG, but our data suggests that although the histone mutation is important, the P53 status is really important. And there's some data coming out that P53 status, I think because 75% of these tumors in patients are P53 mutant, it's been hard to kind of show that that's also important, but I think it makes the tumors even more radio resistant. And I think we also have to tackle P53. I think, our data suggests that P53 is even more important than the histone mutation. The histone mutation is more of an initiator. It prevents the cells from differentiating to become mature cells. So they just stay poised to acquire more genetic alterations. So I think that's like a great question, because I wonder, like, is it worthwhile to downregulate the histone mutation as a new therapy? There was someone at Cold Spring Harbor developed an antisense against the histone mutation. And I'm trying to decide, should we bring that to Sinai and be like the first place that tests this new therapy where we downregulate the histone mutation? Would that have a dramatic effect on these patients or not? I don't know. I guess that's something that I'm still trying to see if we can translate this here, but I'm not sure if it's going to work.
Thank you.
Anyone else? Well, if no other questions, again, thank you. Thanks again. And I look forward to working with you here at Sinai.
All right. Thank you very much.